So I want to welcome you to my favorite models, models that go the extra mile. I'm Chris Herman from 3D Molecular Designs, and together with my colleague Rachel Mosey, and there are a few other colleagues in this group as well. Rachel and I will moderate the session, the chat, and manipulating the slides and things. But what I'd like to do first is introduce you to our presenter, Kim Parfitt. I pronounced it incorrectly again, didn't I? It's Kim Parfitt, correct?
Yes.
All right, and I just lost there we go. Kim has been an educator for 18 years in Cheyenne, Wyoming. She's taught AP Capstone, AP Environmental Science, AP Biology, and other life and earth science classes at the secondary level. She has also been a teacher ambassador and curriculum developer for Howard Hughes Medical Institute BioInteractive since 2014. She's worked with the Nature Conservancy, the National Wildlife Federation, the National Park Service, and the U.S. Forest Service, among others, to develop educational programs. Kim won the Outstanding Biology Teacher Award for Wyoming from the National Association of Biology Teachers in 2017 and the Presidential Award for Excellence in Science and Math Teaching in 2013, as well as other regional awards. Kim has a BA from Michigan State University and an MS in Natural Resources from the Ohio State University. She's a national board certified teacher in adolescent and young adult science, biology, and Kim retired at the end of the last school year and splits her time between Cheyenne, Wyoming and Boise, Idaho. Welcome, Kim.
Thanks. I want to say hello to everybody and thank you for being here today. What my hope is today is to share my enthusiasm for these amazing models that have transformed my teaching. I'd like to identify models that support year-long learning. So when you make an investment in these models, it's not like you just bring them out for one lesson. They are ubiquitous throughout the year. You're going to investigate stories at the macro level that are actually enhanced by the models at the molecular level. And I want you to explore your student modeling pack if you have it. I was wondering if people could somehow communicate with us either through the chat or raise your hand to tell us if you have a modeling pack. So you got it ahead of time by, okay. If you don't, no worries. Okay, we're going to walk you through this. There's lots to do here this morning or this afternoon. So don't worry if you don't have one. But I do want to encourage you to, at the end of the session, they'll explain, Rachel and Chris will explain how you can get your own student modeling pack for participating. Okay, so we're going to move ahead to the tools that we need today. We will be using Poll Everywhere a few times, and that's very simple. The directions will be right on the screen, and it's just a very simple thing to do. You can do it from your computer as a separate tab, or you can use your cell phone. We're going to use chat for questions and or comments, especially if you want to know more about a model real quickly, about how to get it or what kind of versions you can have, like a student pack versus the classroom set. The folks from 3DMD are amazing and they'll be happy to answer your questions. And then also, if you have your double helix student modeling pack, awesome. If not, no worries. All right, so let's go ahead and I would like to share with you. So Chris, we can move ahead, thanks. I just want to say I've been working with the folks at the Center for Biomolecular Modeling and 3D Molecular Designs for probably the last 8 or 9 years. And I am not exaggerating when I say that they have transformed my teaching. This group is, in addition to selling amazing models, they also provide teacher PD in the summer, and then of course through these professional conferences that are so engaging and supportive of teachers. When you go into these PDs with this group, you're not just learning about molecules, you're actually meeting other people that are phenomenal teachers from around the country, like the two ladies in this picture. And then also you're joining a family. and they take care of you. And it is really an amazing community when you start to work with this group. Chris, go ahead and do the next slide. Just to show you an example of how responsive 3D molecular designs is to teachers. When we were hit with all this amazing craziness of the pandemic early on in March, they rallied and immediately developed a series of free videos and some materials that were free for teachers and students to understand coronavirus. And as we move into this next stage of trying to understand all the different drugs and how they're tested and the vaccines, they're still producing those kind of resource materials to help us understand what's going on. So, you know, they really, I'm not kidding you, they love teachers and they support us and they care about us. So I really can't say enough about them. Okay, but you want to hear about the models, so I'll get on here. So let's go ahead and go to the next slide. We're going to use a poll everywhere, and it should be real simple just to go to pollev.com, and you're going to go to Kim P878, and I think Rachel is going to put that up in the chat for everybody. But I'd like you to think about one word to describe what has surprised you most about learning in a time of a pandemic.
Chris Chow says adaptability.
Okay. Yeah, so if you don't mind just putting it into the chat, that way we can see it. And I'm not sure why this isn't working out for me, but you know, that's how it is for technology for me today. So adaptability, yeah. Pivot.
Interaction with my students.
Mm-hmm. Resilience is one that I have been amazed at, not just for teachers, but also for students and families. Exhausting. Yeah, my heart goes out to everybody. All my friends talk about the double duty, triple duty that they have to be doing right now. Patience. Yeah. And pivoting. Yes. We're all pivoting all the time. You know, the learning that I've done in the last few months with this has been amazing. Eager. Yes, I love it. Okay, well, I wanted to just kind of ground us in the fact that, you know, as teachers, we rise to the occasion. And I just am so impressed with people. I know that in California, the next gen science standards are very important to you, as they are here to me in Wyoming. We have a modified NGSS, as you can imagine, with some of the language. However, my district is very committed to NGSS standards. So I wanted to use those standards, the 4 big ideas in life science, to kind of organize the presentation today and to organize how I use the models. So using these 4 big ideas of molecules to organisms, ecosystems, interactions, energy and dynamics, heredity, inheritance, and variation of traits, and lastly, biological evolution, unity, and diversity, I wanted to use those to organize how I use these models. So I also have been using storylining in my teaching, or anchored inquiry, or phenomenon-based. These are some of the stories that I use, and regardless of whether I'm doing storyline or phenomena-based teaching, or whether I am walking through the way some of my colleagues go through those 4 big ideas in sequence, these models apply to all of these, and I use them throughout the year. So 2 model types I'm going to be addressing. First is model kits. These are things that you can get so that your table group or your student pairs can be manipulating the models. And then there's also student standalone models that you can really just have one hanging from the ceiling or on the desk, or just at a station, maybe when you're doing different work in the classroom. So those are the 2 kinds that I'm going to talk about. And when I think about these models, what I love best is that they create what I call that mental Velcro, or conceptual ecology for kids. So even though I might start talking about water molecules and hydrogen bonds way back in August, I'm going to be referring to that over and over and over again. And now my students will have, you know, a kinesthetic experience with these models that then creates that mental Velcro, that conceptual ecology, so that that information has a place to land again when we bring it up over and over again. And that's what's so powerful about these models. Okay, Chris? Okay, we're going to start with the gateway kit, which is the water kit. Go ahead, Chris. All right, so the concepts that I count on. This is, I want to remind students about what a polar molecule is. When I teach general bio, generally these are 10th graders. When I teach honors bio, it's 9th graders. So some of them have not had any physical science for a couple of years. So we go over what a polar molecule is. My covalent bonds are atoms sharing an electron. My hydrogen bonds are weaker attractions between molecules. Adhesion, cohesion is made possible because of that polarity. This is the first time we're going to bring in the fact that there's a color scheme in modeling, a convention that scientists use to communicate their ideas with. This is also possibly the first time I'm going to ask the students to transfer their vision of a 3D model onto flat paper, onto their science notebook. So it's nice to start with this kit because it's very manageable to draw the molecule. And best of all, beginning of the year, I'm going to have them managing materials. So we know how to work with models in the classroom. Okay, so off to the left in my slide, you can remember those 4 big ideas from NGSS. So I'm kind of indicating where in the curriculum these definitely have application. So quick and easy with the water kit is states of matter. So I can talk about kids can model gases, liquids, and solids. And it doesn't just have to be making a representation of that, but they can actually inquire or develop an investigation to show the importance of those structures. Like what is how the density and volume is different for our liquid water than it is in our solid water. And you can take it a step further to start talking about, well, why is ice so important on planet Earth as far as the fact that it floats? And why is that important to life on Earth? So really, with this tiny little molecule, you can teach so many different things. Okay, Chris. Another story that works really well with this kit is transpiration. And so we can show, again, because of the polarity of that molecule, we can show that the adhesion of the water molecules to the cellulose in the xylem and the cohesion between water molecules, like this, allows that capillary action to happen within the xylem of the tree. So students can really get a feel for how this actually works. And model it and make a little video of it, but they can easily do that with these models. Kind of a lesson level phenomena could be the question about, do fish drink water? And so this is really talking about osmosis. And some people feel like, oh, well, it's not really in the standards. They don't say osmosis in the standards, so we don't have to teach it. But I feel like this is such a lovely story about structure and function and about cause and effect. And so I love to do this with our water molecules because it lets the students actually investigate the phenomena and then model it and then create an explanation. So, of course, the answer is, do fish drink water? Well, it depends on the fish, but our freshwater fish, you can see student modeling up on the left-hand side of the screen. With our water molecules, there is more solute inside the fish, and so the water goes into these are bull trout right there. And then in our saltwater fish, our marine fishes, there is more solute outside of the fish, and so the water travels out of the fish. So they actually have to have strategies that conserve water. But my favorite is the next slide. is the hagfish, which is a very horrifying organism that looks like it's from a Steven Spielberg movie, because it has a mouth like a cookie cutter, and it basically is a scavenger and eats dead things by twisting. And they're isotonic. They're in very deep parts of the ocean, and so they actually have equilibrium with their environment when it comes to water moving in and out of their cells. So students can model this isotonic situation. So even if they're not using language like hypertonic and hypotonic and isotonic, they can still show the condition that exists for these fish very easily with these models. I know I was taught, and I think I taught for years, that, water, it's pretty important to us, so it just goes right across that cell membrane. Well, this is a beautiful story of the nature of science about our understanding. And I don't want to steal the story from the other people that will be talking about water, but there is a protein transporter that is the aquaporin that allows water to move through the phospholipid layer. So this is a nice story. If we go to the next slide. What I'd like you to do is look closely. The gray kind of blobby things is representing the aquaporin protein channel. And then, of course, we have our red and white water molecules. So look closely about what's happening here and use the chat to describe what you notice happens to the water molecules as it passes through the aquaporin.
Laura says the hydrogen atoms change direction as they pass through.
Well, I see single file from Maureen.
Specific orientation. Oh, Karen says, I see charges.
Yeah. All the red point one way. Yeah, everybody is seeing a really unique situation here. I just love this. I want to stress that there is a wonderful video animation on the 3DMD website that you can use with your students to actually show this in motion. But everybody's starting to see what's happening here. So let's go to the next slide. This is an actual aquaporin model from 3D Molecular Designs, and you can see that inside there, it's showing you the channel and then the water molecules inside the channel. But there is a side chain, and that's what Karen had noticed about the charges. The asparagine side chain actually has a positive charge. And so when it narrows, when that channel narrows, the water molecule, the oxygen is attracted to that positive charge, and it kind of ends up flipping, as somebody said, it's kind of flipping or arranging the molecules so they fit through, so that by the time they get to the other end of the aquaporin, they're actually oriented in a different direction. So if you go to the next slide, yeah, the molecules rotate. Good. Yeah. So we can see that here. And again, it's that polarity that is, the polarity is what's kind of a challenge, could be a challenge to go through the phospholipid bilayer of the cell membrane. So the aquaporin is an elegant solution to that issue. All right. So aquaporins might be pretty important when it comes to life on Earth. And as a result, we can actually talk about how there are different classes of aquaporins in plants and organisms and animals. And you can talk about how these are very highly conserved genes because they stick around because they work. But also the different classes, and by investigating them, you can actually look at relationships between organisms by looking at similarities and differences in aquaporins. So there's a nice opportunity to look at evolution when it relates to how does water get across our membranes. Now, this kit is very rich, as all other kits are. I haven't even touched on some of the things that this small kit will do. For example, one of the things is it will talk about solubility by providing you with sodium and chloride. And kids love this. because it clicks. And so you have to watch these because these have a tendency to migrate out of the classroom because they are very, very fun to play with. Something that I didn't show you in the beginning that I wanted to show you is another really elegant way of getting kids to understand the difference between a covalent bond and hydrogen bond. And that is in the molecule itself, my hydrogen atom will come off. I can pull that off, but it takes a while, right? It takes some energy to disrupt that covalent bond and pull that off. But when I have my hydrogen bonds between my hydrogen and my oxygen, it is an attraction, right? And it can be broken and reform, which is one of the, magic things about water to begin with. So that is a really nice takeaway for students. And my students remember that for years. They come back and talk to me about how they understand bonds better because simply playing with these molecules. All right, when you go to the 3DMD website, you'll see, go ahead, Chris, that with this kit, there are a number of pre-made lessons, both resources for teachers as well as worksheets for students. And I know a lot of teachers are very concerned about students finding answers on the internet, but they actually are locked away. You have to request answers for the student worksheets. So you can count on them if you are somebody that likes to use those for graded assignments. So another nice thing about this is, regardless of how you're teaching, whether it's virtual, whether you're doing the hybrid model, or whether you're in the traditional face-to-face, 3DMD has a variety of materials and resources for you. So one of the things that, go ahead, Chris, is in addition to our student modeling packs, which this is the double Helix student modeling pack for this class, for this session. But for all of the student modeling packs, they have free digital activities. So, yeah, Chris, if we could click on that and get that little interaction moving. So even if students don't have this kit at home, it's okay because they can still access these free activities. They can interact with it. And here's the nice part is that there are questions that are like active fields. So kids can type in their answers or their reflection about what they're doing, and they can save it as a PDF and upload it to your learning management system or email it to you, whatever you'd like to do if you want to see their work on these. So again, for every single student modeling pack that's offered, they have one of these just terrific digital activities that students can use regardless if they have the pack or not. Now, let's say that I am face-to-face and I would manage the materials in my classroom. In action, when I'm using these with my students, I have 1 cup of water per group of 4, but it actually even better is to have 1 cup per pair of students. I use a cafeteria tray or even an upside down Frisbee is sometimes what I use to manage the materials. Otherwise you get water all over the floor and they bounce. The kids think that's pretty fun at first, but they realize then they can't keep track of their models. So having a little tray of some sort really helps. Later on for hanging around because the story and this model becomes very important for that mental Velcro, that conceptual ecology, is they're magnetic. So I keep a couple on my whiteboard so I can just grab one and say, hey, so now you remember, tell me about what it means to be a polar molecule. And again, when they get to this very remarkable little model, they're able to remember. And plus, in the beginning of the year, you can have lots of water jokes like, oh, it's raining, and you have the water molecules fall apart, or watch out for the water on the floor, so they like that. All right, so that's the water kit. Very tiny, incredibly useful. So let's move on to the next one. This is the amino acid starter kit. And honestly, I call this the magic aha kit because it is exactly that. What used to take so long to teach, right? The central dogma going from, DNA to RNA to a protein. I remember when I first started teaching biology, it would take 2.5 weeks and we would do endless rounds of, transcription and translation. And Oh my gosh, it was brutal. Kids didn't see the point. 2.5 days with this kit, and my kids are like, they are masters and they love it and they feel efficacious with this. So what I count on with this kit, go ahead, Chris. Oh, let me back up for a second. I like to preface using this kit with these little building block models, the amino acid building block models. They are what my kids and I refer to as a poodle on a Segway when they're built, because that's kind of what they look like. But this little model allows students to have in their hand a very tangible representation of the amino acids, so they can see that NCC backbone and they can build a polypeptide chain. And when they build that polypeptide chain, what you see there in that photo is the resulting water molecules as a result of dehydration synthesis. And then, of course, when we break apart our amino acids, we're doing hydrolysis. My AP Bio students never got those two mixed up once we started working with those little tiny poodles on a Segway or the amino acid building blocks. So once I had worked with that with my, kind of when we do macromolecules or talking about how molecules are utilized in a living system. Then when I got to genetics and DNA and the central dogma, they already had some familiarity with that NCC backbone, and they knew that there was a side chain. But now with my amino acid starter kit, now they start to see that amino acids are the same when it comes to that backbone, but the side chains make them different. The folding of the protein is determined by 4 rules. The primary structure of a protein is a strand of connected amino acids, and that the order of the amino acids on that strand is determined by DNA. What I love best, these are the Kim Parfitt personal cross-cutting concepts of complexity doesn't have to be hard and biology is beautiful. And that's what I love about this kit. So students will have an opportunity to work closely with amino acid side chains and then actually building a folded protein. So with the amino acid side chains, what I love here is students are actually looking for patterns on the arrangement of the atoms that help them determine the function. And so this little pizza wheel is what we like to call, actually helps them organize around those ideas of how the amino acid then is going to behave because of the structure. So those will come in handy when we talk about our 4 rules. So there are our 4 rules. We have our primary structure up at the top, our polypeptide chain that the students will build using a tuber. And then we have these very 4 simple rules of chemistry. The hydrophobic amino acid side chains will want to be on the inside of an alpha helices. And the next one is our positive or negative charges will want to attract because opposites attract. We have our cysteines that create a cysteine bridge, which provides a lot of strength to a molecule. And then lastly, our hydrophilic amino acid side chains will want to be on the outside of the tuber or the molecule. So once they understand those 4 kind of playing rules, then they can build a folded protein at the secondary structure level and the tertiary structure level. And of course, you can bring them together to do the quaternary structure level. So once my students start to see the relationship between the amino acid side chains and then the actual structure of the protein, it's like a little light bulb goes off. And then we can go on all sorts of directions. So literally that takes me 2 class periods, and my kids are just feeling like they really get excited about proteins. Now we can go into all sorts of different directions. One of my favorite things is to, of course, link to who they are, link it to their bodies. And of course, some of them still watch Big Bang Theory and they know Sheldon. And so we can talk about proteins and what happens, how they can be denatured when we have a disruption of our acidity in our bloodstream. So we can talk about pH. I can bring in my water molecules and actually have my students demonstrate this if I want to. An obvious one for me is to then take the next step where we can start talking about sickle cell anemia, and we could talk about hemoglobin. So we want to talk about the single nucleotide substitution that results in a change in the shape of the molecule. And so I bring out the amino acid starter kit again, and they use the first 16 nucleotides to actually determine the primary structure. And then they see that they're going to actually switch out glutamic acid for the valine. So Chris, if you want to hit that again, there we go, thank you. A secondary model that I bring in here is the hemoglobin gene map. And this is like a 9-foot long laminated piece of paper that is actual hemoglobin B genetic information. It shows not only the DNA strand, but also the amino acids. So the students can actually learn about exons and introns. They can actually point to the mutation that is creating the sickle cell anemia. And then later on, when I start talking about evolution, we can talk about heterozygous advantage, or we can talk about hemoglobin in other organisms and how we can determine ancestry and relationships between organisms based upon those structures. Again, if you go to their website, I am, of course, not even coming close to all the things that this kit you can teach with, but it does list it at their website. And once again, if you go to the site, you'll see there are lessons for each aspect of the kit, so that you have teacher support materials, you have student worksheets, and because 3DMD loves teachers, now you have the student modeling pack for the amino acid starter kit, and you have these free digital activities. So if I'm face-to-face, how I manage these materials in my classroom is I use one amino acid starter kit per group of 3 or 4 lab groups. And then I instruct the students to only take certain things out of the bag at a time. It is a complex kit as far as it's got a lot of parts to it. And so we try to, make sure that kids aren't getting out things that they don't need at the same time. And then they focus on things at a time. There's a lot of their amino acids are fairly small, so it's easy for them to get hidden under a piece of paper. What's nice about this kit is the kids, it's such meaningful learning for them. They want to take care of it, right? I have used these kits for years now, and I have never lost any pieces in my amino acid starter kit. And it's because the kids are very conscientious about putting things back together, putting them back in the bag, sealing the bag up, making sure that everything's there, because they know that we're going to use them again, and because the learning is so valuable to them. If I'm just wanting to make sure that I have this to point to or to pull up off my desk, I always have I can magnetize the pizza wheel. Actually, the old versions were magnetic, and I keep that pizza wheel with some side chains up on my side, my whiteboard. But also, I always have one of those tubers folded on my desk, because I'm always holding up a tuber and we're talking about the denaturing of this because of pH change, or we're talking about how the side chain will do this, or how the alpha helices will work, and things like that. So that tuber is really kind of a mainstay on my front desk in my classroom. All right, so the next one, the DNA starter kit. And this one is so accessible to students. So this is going to be your turn. I think what we're going to do here is I'd like to introduce some of the logistics of this kit, and then we're going to give you some time to work with your kit. And for those of you that don't have the kit, Heather is going to actually put the kit under a doc cam so that you can see all the parts and we can play around with that. Okay, so let's go ahead and go to the next kit, or the next slide. All right, so these foam models will have, as you can see, it's like a puzzle piece where you've got you've got like my thymine here, and it has these arrows that are hollow. That's the receiving part of the hydrogen bond. For my adenine, there's the hydrogen bond, so that when they come together for their base pairing, they fit together like this. Now, 3D-MD, I'm telling you, they think of everything. So they've provided you guys with this little baggie of these adhesive bands. And the adhesive bands can go on the thymine and the cytosine as a way to provide extra oomph for when you have your base pairing. The directions tell you to put both bands on, so it's almost like this becomes like a little sandwich. I don't do that. I gotta say, I don't do that because I like the kids to be able to see the hydrogen bonds. And then also this is kind of sticky. And so that's why this white powder that you can have, flour, cornstarch, talc, anything like that will work. And that's where you're just going to dip the prongs on the guanine and the adenine. You just dip it in the flour so that when you go to put it in to the hydrogen bonds on the cytosine in this case, it doesn't stick and it won't rip the foam. So you can do that. And that, like I said, it kind of gives it a little extra oomph when you're messing around with these. I, in my classroom, have never done that trick. I have always just left them like this. And so far, I haven't had any problem with them popping out or being too loose. So I think you would need to use your judgment with your students. You know your students best and how often you use your model. So let's go ahead to the next. So does anybody have any questions about that kind of prepping? If you could throw that up in the chat, we can answer that. But so there's 3 options there. You can do a double-sided band, you could do a single-sided band, or you could do no band. It's really, really up to you. So let's take a few minutes. We're going to take about 5 minutes on the timer here. I want you to play around with your model if you have them. And I want you to play around as if you are a student. You're going to have your gray, long, skinny sugar phosphate backbone. You're going to have your nitrogenous bases. And I want you just to go ahead and build what you think you can build with this. And I want you to kind of keep track of what do you notice and what do you wonder. We'll use the chat to share out 1 or 2 noticings when we're done at the end of 5 minutes. If you don't have your student modeling pack, no worries. Heather is going to show us using the dot cam how some pieces of this kit. So are we good, Heather?
Yep, I'm gonna play a little bit here. So if you have any questions or want to see something again, just shout out.
Okay, so Heather, can you tell us what you're doing there?
Yeah, so right now I'm building a DNA, my DNA model. I am base pairing here and adding them to my backbone as I go. It's probably a little bit more correct. I don't know. Yeah, it's probably better to go this way, huh?
Well, what I like is for those kids that have had some experience with DNA, you'll see them quickly, immediately go to that base pairing, right? They understand the A to T, C to G. They see that, they remember that. Another thing that I like that Heather's doing is she had the one side of the DNA molecule created first, and then she built the second side. A lot of kids will put their base pairs together first and then put the backbone, sugar backbone on. So that's an opportunity to talk about, well, what really happens in the nucleus of the cell when this is going on. Okay, so then you can see the flexibility of the model. And that's where when they're doing that twisting, if you have the little bands on the cytosine and the guanine, I'm sorry, on the thymine and the cytosine, then it actually kind of helps maintain that accordion-like structure.
As we were trying to think about what we could do to get this in the hands of more students when they couldn't share materials, what I realized this this summer is if you take one of these gray backbones for sugar phosphate pieces. So if you take this, and then if you just cut it in half, so this is what I did here. So I have one piece, this is my solid piece. Then I just cut it in half. And then that let me make 2 little 6 base pair models. So then each student could have this model instead of having a 12 base pair model.
Nice. So students can still manipulate it, but they don't have to share materials. Good.
Yes. Mm-hmm.
Okay. Laura, you asked about, do they rip easily? I would say not really. I have had mine I've had my kits for probably 7 years, and they have not ripped. They're durable.
And we did tell people that they can register online and they can get a free double Helix student modeling pack in the mail, right?
Yep. You know, and I will say that sometimes when I went to the PDs in the summer, and I might have gotten one of the kits or one part of the kit, I would use it as a station when I was doing some independent learning with students, or just having it up on my desk, keeping it up there still was engaging for kids. Kids would come up and just visit with me. Sometimes when kids would come in at lunchtime, they would want to play with the models and just visit. So they're so accessible. Whoa, okay, time's up, woohoo! Thank you. All right, so if people could actually put in the chat maybe something they noticed as they were working with their models and that they were building their model. And then also, Chris told me that I think we're allowed to ask you if you would like to show what you built. You can show your camera and hold this up in front of the camera so that we can see what people built. That would be awesome.
Great. Hello, everyone.
So one of the things that happens is, you know, the kids love doing this. They love doing the accordion. And then also you can ask students, you hold them up and then they ask, you know, are they exactly the same? Did everybody come up with the same sequence? What was different? So you can start talking about the sequence of the DNA and get that word sequence in so they can begin to understand when you get ready to talk about what a gene is. You've already discussed the sequence rather than base pairing, because they love the base pairing. It's something they can pair it back. But the sequence part sometimes is a little elusive. Do you do a variation to show the building of the backbone? Joseph, can you give me more information about your question?
Yes, I have. I'm grabbing some other pieces. So the DNA starter kit. So this isn't the student modeling pack you guys are getting, but this is the regular classroom kit that we sell. So it comes with both continuous backbone pieces, but then we have some individual sugar phosphate backbones. So here's the nucleotide attached to an individual sugar phosphate, and here's another one. So as you are, that was a triphosphate, we won't get into that.
Yeah.
But you can actually have your, you can build the backbone this way.
Right, yeah.
Wait.
So Joseph, that's a great question, and we'll show you that's what allows the kit to actually do replication. And so the kids can see the semi-conservative nature of DNA when they use the DNA starter kit. But like Heather said, that's not part of the student modeling pack, but it is part of the DNA starter kit that you can get. So
I don't have more of these handy, or else I'd show you.
okay, you keep going. Well, I've got that in the slide.
Yeah. Okay.
All right. So one of the things the kids notice is that there's arrows on this, and that this is pointing, it's a one long arrow. And so then you can actually have a conversation about the anti-parallel nature of DNA. And kids they're very curious about this. And then also just the shape of the nitrogenous bases can get them thinking about, well, what is it that allows this molecule to store and transfer information? And so this is an example when, after I have my kids kind of messing around like we had with you guys, just mess around with the kids. And that lets me see which kids remember things from 7th grade, which students don't remember things, and they're just kind of moving along. Then I actually have them in their science notebooks create a notice and wonder chart. And that helps me, again, it's kind of like a little pre-assessment before we start actually kind of digging into DNA a little bit more. Go ahead, Chris. The first thing you can teach with this, and it's so natural for the kids to see the base pairing. My kids come in knowing A to T, C to G, but they don't know why. They don't know why they go together. And so this lets them see that, right? Because the hydrogen bonds right there allow them to understand why these molecules go together like this. So again, that structure and function piece. So the accordion nature, they love to do that. And that gets into the whole how this huge long molecule can compress down into this teeny tiny space. And so they can do that. And that's very satisfying for them. They love that part. Okay, Chris. And then another thing I love to do is, and this may seem a little deeper for maybe Gen Bio, but I still do it because it helps with organizing patterns that they see. And that is to set up a little table that shows how many bonds and how many rings. And so when I do it this way, then I can see that I have my purines in my first column, and my pyrimidines in my first column, and my purines in my second column. So they can start to see that it matters what these molecules, how they're shaped. So then we add them up. So I count across, you know, how many rings on between adenine and thymine? 3. How many rings between cytosine and guanine? 3. So now we go back to our accordions and we see that compression. Chris, go ahead and go to the next slide. And I have all the students look down like this so they can see that that equal width matters. Okay, my molecule is not going like this all over the place. It's not, it's very predictable. It's all the same width. For those of you that might use some of the materials from Biointeractive, the double helix film has a great component in it where they're actually visualizing these cardboard paper models and how they go together. And it's a beautiful complement to this model. And the kids can suddenly, that becomes so valuable to them to know that they were experiencing the same kind of modeling that Watson and Crick were. Okay, go ahead. The next thing I do is I make sure that my students can transfer the information that they have from this simple model to more complex models. And so this is another resource that's free on 3DMD's website. It's could be a coloring sheet, so they could see that, again, that CPK coloring scheme that scientists use kind of as a convention in models. But also they can show me, okay, where on your foam model do you see hydrogen bonds? Where on this structure do you see your hydrogen bonds? Where's the sugar? Where's the phosphate group? Where's the nitrogenous base? So this is just an opportunity for them to practice transferring that information, right? And this is where we have replication. So I know Joseph had asked about the, do you have a way of building the sugar backbone? And so here, these girls are starting to build the, they've unzipped their DNA molecule, and now they're building the complementary strand and using the sugar phosphates that come in the DNA starter kit that Heather was talking about. Okay. Now, again, in the double helix student modeling pack, this is kind of an abbreviated version of our starter pack. And when you get the starter pack, you'll also get uracil. So now you can actually begin showing about how to build mRNA. So if you wanted to talk about transcription with this, you could. Okay, so this is a picture of my classroom. The students have their student modeling or their DNA starter kits, but there's one model that's different. And I pulled that off to the side. This is the dynamic DNA kit that was the session this morning. And this is a kit that, again, is like knock your socks off kind of kit. And it makes things so accessible to students. So this was an opportunity in my classroom where we were using multiple models so that we could actually evaluate models, right? What is the strength of this one? What is the weakness? Because when we make models, we always know that all models are wrong, but most models are useful. But when we make models, we have to make decisions about what is important. And so this is part of the science practice of communicating our ideas. And so we have to actually determine what message are we sending, or what is the key information that is most important for an observer to see in a model that we're working with. And so when we use multiple models, it really gets kids thinking about kind of the grain size that they are using when they're creating an explanation for a phenomenon. Okay. So again, you go to the website, and there are worksheets for teachers, worksheets for students, directions, animations, all sorts of resources to help you utilize this kit to the fullest. And if you are, of course, just like all the other student modeling packs, there are digital activities students can use. Even if they don't have the kits at home, they can still use those activities. So if I'm face-to-face in my classroom with my students and we don't have a pandemic going on, I use one kit per group of 4, and within one kit I get a number of these backbones and enough of these for my partner groups to actually create their own molecule. I think, what did you say, Heather, that this is a 12 base pair molecule? So, but I loved her idea of, you could cut this so that kids don't have to share materials if that's something that you can do. Hanging around, again, this is something I keep on a shelf. Kids can see it when they walk in. It's easy for me to grab. So if I want to demonstrate something, I have it out all the time. In fact, all of the models that we've talked about so far, they are on deck at all times. All right, so let's go to the next slide. So here we're going to take about a 7-minute break so that we can do what we need to do, get a drink, get a snack, get a drink, stretch your legs, and we're going to come back and we're going to talk about our next model that includes some death and toxicity and trickery. So we'll see you in 7 minutes. All right, so I hope folks have gotten what they needed, and we will continue on. If we want to, if I could just go back to that scorpion slide for a second. Before I move on to this next model, I just wanted to address Joseph, if you're still with us. You asked, and I know Diane had talked about, yes, do whatever you need to do to your models and that you can write with a pen on the foam. For like 3 and 5 prime on the sugar phosphate backbone. I use stickers, just the little colored dots that you can get from an office supply store. I use stickers on the backbone. And then also when I use the chromosome models, I use stickers to show alleles. And also the reason why that showing multiple models at one time is great is because then you can discuss how, like on this one, our sugar phosphate backbone is really representational, just here to suggest that we know it's there. Whereas in the dynamic DNA kit, it has the atoms can be shown. So you can actually see the directionality on that model, so that kids can understand what's happening with the actual structure of the oxygen. So you can show, again, that there's decisions that have to be made when you're working with and making models. Okay, so death, destruction, toxicity, let's go for that. Let's talk about this one. This is a standalone model that I would consider, although certainly if you have grant money or you have a nice budget, it'd be great to have one of these for every table or lab group in your classroom. This is a two-part model of a potassium channel and scorpion toxin. And so the green model and the little red circle that just popped down there shows you the potassium channel that allows potassium to go across the cell membrane. And there's 4 little oxygen molecule or atoms right there that help help the potassium travel through there very rapidly. Now, this is important because this is what we need for things like moving our muscles, some other things that go on within our cell processes that are critical for living. And so we want that to be open and happy. And so those potassium ions can go right through either way of our cell membrane. The scorpion toxin is a simple peptide that actually has a structure that prevents that from happening. So if we go to the next slide, you can see that the scorpion toxin, in addition to having a number of cytosines, and kids will remember from our amino acid starter kit that the cytosines create that disulfur bridge that is very strong and provides kind of a structure. And so that's what happens with our scorpion toxin. There are 8 cytosines that make 4 bridges. And then we've got a lysine that has a positive charge. Now you can ask your kids, if this was positive, what do we know about our oxygen? Well, we know oxygen has a negative charge. And so what do opposites do? They attract. So then we see that our scorpion toxin can dock right on the top of the potassium channel and block those potassium ions from going into and out of the cell. And that is what causes paralysis. And then the scorpion, if you are not the size of a human, can eat you and carry you away to feed its children. So let's go to the next slide. Let's think about the structure of these proteins and how they're interacting. So what might prevent the toxin from working? What could possibly prevent this toxin from working? So let's give me an idea in the chat. Now that you think about that protein and how that lysine fits right there to block that channel, what is something that might happen to make that not work anymore? So go ahead and give us some ideas in the chat. Oh, Laura, I like this idea. Could there be a block? So could something block the scorpion toxin? That could, that's an idea. So when we think about the fact that we have this folded shape of our potassium channel, if there is simply a different shape, that could act as that blocking of the toxin from having the lysine be able to get in there and block that that channel. So a simple change in the DNA sequence in the gene would change the sequence of the amino acids, and then that we could change the actual shape of the protein. Well, that is exactly what happens in the grasshopper mouse. So Maureen, yes, that's exactly right. So the grasshopper mouse is a warrior mouse. It's one of the only rodents that actually eats meat. And so we're gonna watch a little bit of this National Geographic video. I will warn you that there is some carnivory going on with our mouse, but we're only going to watch about 1.5 minutes, Chris, so that we can see it attacking the scorpion. We don't need to watch about the centipede. We're just going to focus on the scorpion.
In the middle of the Sonoran Desert, this mother grasshopper mouse tends to her pups. While tiny and adorable, don't be mistaken, this is not your common house mouse. From the day they are born, these mice are natural killers. Insects, lizards, scorpions, and even other mice are all on the menu. This mouse is distinguished from most other rodents in its reliance on a meat-based diet. It regularly takes on prey as large or larger than itself. Making the grasshopper mouse even more of a standout is its immunity to scorpion venom. Not only can it withstand the typically deadly stings, its body has adapted to convert the toxins into a painkiller. After a scorpion, a giant centipede might seem.
All right, so the nice thing about this story with the mice is that I can actually, so now I'm taking the molecular ideas and transferring it into an organism, and it actually can go discuss about evolution, and then I can also talk about populations. The mouse has some really unique behaviors like howling to set up its territories. And so this can be kind of, again, another little lesson level phenomena that can really generate a lot of student questions. And the models can actually help them then generate an explanation. All right, so the last kit that I'd like to talk about today is our phospholipid and membrane transport kit. Again, there is a student modeling pack, and then there are classroom versions of this. So go ahead, Chris. The concepts I count on with this is the hydrophilic and hydrophobic interactions that happen with this molecule of the phospholipid and within the membrane itself. Bonds versus attractions inside and outside can create a gradient. And kids get very excited about when you have inside versus outside. And so the ability to do work when those molecules can move from one side to the other, of course, is our precursor to a lot of things in cell processes. That a cell membrane is more than just like a shiny bubble latex balloon, right? So we have a mosaic of a membrane rather than just a single kind of bubbly layer. And then embedded proteins, the right tool for the right job. And so we can look at passive as well as active transport. So with the phospholipid, you can have kids building the phospholipid to understand this molecule a little bit better, to understand the hydrophilic and hydrophobic ends. You can also have kids doing thought experiments and hand experiments with the small these tiny little phospholipids that allow students to understand how they actually organize in a watery setting. So then you can start getting into, you know, how do cells start? How do we get these cells? And so you can actually work with that. There's a fabulous lesson that walks you through this with kids to ask some really juicy questions to get them thinking about this. And what I like about that is that that kind of questioning they'll carry with them throughout using this kit along the way. All right, so there's our aquaporin right there in action. So there is our passive transport, our aquaporin. You can actually have the little molecules of water do that flip if you want to. If you go over to the right, you can see the small phospholipids. You can show the bilayer and the charges that are down on the one below. And then over to the left with the blue background, we're showing a sodium potassium pump. So we're showing active transport. All right, so active and passive transport could be part of our story with photosynthesis, right? Because my guard cells use both active and passive transport to pump sodium or potassium ions across the membrane. Water follows. We get that turgid or flaccid guard cell that either opens or closes the stoma, allowing to conserve water for the plant in times when it's windy. Like today for me in Wyoming, it's 65-mile-an-hour winds. My plants definitely have their stoma closed or open when they want to photosynthesize to let CO2 in. Okay. Another part of this is these models. This is a student using the membrane model to actually augment her understanding of cone snail venom. And she brings in addition to the model itself, but she brought in some other parts, other pieces, so that she could make the model her own. And then she actually created an explanation with her diagram over here about how the cone snail venom works on the fish's neurology. So they're making sense through these models. They're not just moving parts and pieces around on the table. Again, lessons, if membranes aren't your passion, there are lessons that walk you through all the different things that can be revealed and uncovered through working with this kit. And there are teacher versions and student versions. And there is a student modeling pack for membranes, as well as that online digital activity. And in my classroom, these are pretty big kits. And so I have those big black tables that we have two pushed together, and that works really well. But sometimes I actually end up having the kids work on the floor because the membranes get really big and we can merge our membranes together. But I use one phospholipid membrane kit per group of 4. And, but when I started, I just had one, and I used it as an example. I used it as a station. You can magnetize the back really easily by picking up those magnetic sheets from Michaels and cutting them off, sticking it on there. I can stick it up on my whiteboard, and, or using Velcro if I don't have a whiteboard. Now there's one thing in this kit that's not in this kit, but it's a great complement and standalone, and that is the giant phospholipid. This is Dr. Carrie Shingleton, one of our esteemed colleagues, and here she is holding the giant phospholipid. What I love about this is, first of all, it's big, chunky molecule, and the students love handling it. They can see the carbon and hydrogen and in the hydrophobic tail, and then they can see the atoms up in the hydrophilic head, and they can also see the double bonds that are present. So that can get into the really nice interaction that kids are always surprised about, is that in the membrane, those phospholipids are not covalently bonded together. It's their attractions. And that double bond in there helps create that wiggle room, that space, so that other molecules like cholesterol can fit in there. And that attraction is what makes that dynamic fluid nature. So this makes it very, very easy to see, very real for kids when they get to hang on to one of these chunky molecules. These are great to hang from your ceiling as well, so kids can see them. All right, so the kits that have been my favorite today that we've talked about: the water kit, the gateway kit, the amino acid starter kit, what I consider the aha magic kit. And I also like to pull in those little amino acid building blocks. Inexpensive, so usable in the classroom, and big and chunky for the kids to use. Double Helix student modeling pack, or in my case, I also have the DNA starter kit for my students in my classroom. The scorpion toxin and potassium channel model, which can be just one. I can have one, and I hold it up, and I pull this out all the time. And what I love is that when my kids, they tell other science teachers about this scorpion toxin. So my colleagues know exactly where I am in my curriculum throughout the year, because kids start talking about scorpion toxin and the grasshopper mouse. And then the phospholipid and membrane transport kit. All of these have certainly transformed my teaching and the learning in my classroom. And I always love it when my students come back from the University of Wyoming to talk to me, and they're like, "Why our professors don't have those cool kits?" And they love those kits. So these kits easily embody all of the cross-cutting concepts and the science practices. And of course, throughout the, I think you can see that the 4 big ideas are well represented in this, in the use of these kits. So here's a little video that shows some of the student modeling kits. So the protein student modeling pack, the membrane modeling pack, the nucleotide. Oh, these are great. This really helps kids. No more paper cutting out of A's and T's and C's and G's. Double helix, chromosomes, and the water. And of course, for all of these are the free digital activities online. Even if the kids don't have the kits in front of them, they can still utilize these materials. So thank you for attending. Thanks for asking questions and adding input along the way. I appreciate it. And if you have any further questions, I hope you'll stop by the booth and play around with some of these models.